All right, time to forge ahead in our discussion of our introduction to microbiology. Today's topic is biotechnology, chapter 9, biotechnology and DNA technology. There's a little beautiful micrograph, false color micrograph of a human immunodeficiency virus particles budding off from a host cell, probably a CD4 positive lymphocyte. All right, biotechnology, what is it? It's the use of microorganisms, cells, or cell components to make a product. In other words, we're going to harness the equipment of certain cells to make something that's useful to humans that they didn't normally make. And how are we going to do that? We're going to do that with recombinant DNA technology. We're going to take genes that we're interested in the product of and insert them into the genome of another organism and form this hybrid, this recombinant DNA in a organism that can then produce something for us. A vector is a self-replicating piece of DNA that we can use to carry a gene into the new cell. We said that we want to take a gene from one organism that we're interested in having the product and insert it into another. Well, we're going to use a vector to deliver that gene of interest to a new organism. Typically, the most common types of vectors are plasmids, just a double-stranded circular piece of DNA. And plasmids, as we'll see in a moment, normally occur in bacteria. They were discovered as little riders inside of bacteria, and they've been isolated from bacteria and modified in just an unbelievable number of ways to produce all kinds of variants that can be used in the laboratory to do just a myriad different number of tasks. Also, viruses can be used as vectors very often. So we can engineer a virus to carry a gene that we introduced into it and then deliver that to, especially to some eukaryotic cells. And then those eukaryotic cells will produce a product for us or be altered in some way. A clone is a population of cells arising from one cell. So let's say we insert a gene of interest into a plasmid, and then we say, wow, we need a lot of copies of that to do something with it. We can introduce that into a bacterium and then let individual bacteria that now have that plasmid in it grow into colonies. And each colony is called a clone. It's a population of identical cells that came from that first cell. So here's like a general scheme of recombinant DNA or biotechnology in action. So here we have a bacterium that harbored a plasmid and say, well, we could use that plasmid as a vector. So we'll purify out that plasmid by growing a bunch of that bacteria, a clone of that bacteria, bust them open and purify the plasmid out. So that's what we have here. And also I should add, as I mentioned, modify that plasmid to make it much more efficient, to have all kinds of properties that make it even more useful to the laboratory researcher or the industrial researcher. Then there might be a gene that we want to express, something we'd like to have the product of. Let's say it's an insulin gene from the human genome. There's multiple ways we could get a copy of that gene. One way we can do it is cutting up the genome of the species of origin using some enzymes that can just break it into manageable pieces and then insert those pieces individually into a bunch of plasmids. So take a whole mass of like thousands and thousands, millions of plasmids, cut the genome up into thousands of pieces, and insert all those pieces into individual plasmids. Then we can introduce all those plasmids that now have a piece of the original genome, including at least one that has the gene of interest in it, into bacteria, and then grow clones of all the different possibilities, all the different bacteria that have different pieces of that genome, and then we can screen and find the one that has the gene of interest, and then we can create clones of that and purify it, either purify a product or something like that. It might be insulin. It might be a blood coagulation factor, a plasma protein normally made by the liver. Now we can make bukus of it, buckets of it in the factory in a great big vat of bacteria that now have that particular gene. It might be a bacterium, it might be an animal cell that we're using to produce proteins like clotting factors, antibodies. Vaccines, sometimes vaccines are just protein products that are from a particular virus, for example, and we've inserted that gene into a bacterium or into an insect cell or a mammalian cell, and now those cells will produce that foreign protein. We can inject that into people's bodies, and their bodies will make antibodies. The humans will make antibodies, so we have vaccines. All kinds of good things can happen. So we either can create a large number of bacteria containing the plasmid with a gene of interest to use in the lab for something, just to have copies, or we can force bacteria or animal or insect cells to make the protein product or an RNA product for us. We know that RNA vaccines are a big thing. In this past episode of COVID pandemic, what do we do instead of making proteins that represent a product of the virus? Made an RNA that represents the sequence from the virus, and then let the human cells, the vaccine recipient cells, make the protein and then form antibodies and so forth. So that's the general scheme. Plasmid is a vector, a gene of interest, combine them together, uh, amplify them, get a whole bunch of them made in bacteria, and then use those to either infect, either grow up a bacterium that's producing a protein, or introduce the gene from the bacterium into a animal cell that can make an interesting product for us. Growth hormone, the picture here is showing a little boy or girl who is behind statistically the children of their age in size and stature. And they might be injected with some human growth hormone in order to help speed up their growth a little bit, their long bone growth, and get them a little taller. Here's a list of some prominent examples of where biotechnology has been used to produce pharmaceutical products, medicines essentially. I just picked out a couple to throw that you might have heard of that throw out there, but you can read this list. Maybe some of them you know about or find very interesting. Cervical cancer vaccine, human papillomavirus vaccine. You've probably heard of the HPV vaccine. Erythropoietin, you've probably heard of blood doping. Well, sometimes patients need a little shot in the bone marrow in order to get more red blood cells made. We can have some recombinant erythropoietin to inject into the patient, and then their bone marrow will produce more red blood cells. Hemophilia, people that are born with a genetic anomaly in which they don't produce one of the blood clotting factors, they have a bleeding disorder right from birth. And so that used to be a death sentence, and now we can just go to the factory, produce a large amount of the missing protein clotting factor, and inject that into patients periodically, and they can have a normal life. Wonderful breakthrough. Hepatitis B vaccine, if you work in a clinical setting or plan to work in a clinical setting, you either have been offered the hepatitis B vaccine or had the hepatitis B vaccine or will be offered. It's a hugely prevalent vaccine, very, very important one for healthcare workers, and that's a product of recombinant DNA technology. Insulin, diabetes mellitus type 1, people that can't produce any insulin. Again, a horrible disease, but now relatively easy and cheap to make. Well, it was cheap to make insulin and provide that for the patients. There's been a problem with insulin production and insulin prices lately. I don't know exactly what that's all about, but I'm a little cynical, but I think it's not good reasons. But anyway, it's a great breakthrough that we can make insulin to provide for diabetics. Influenza vaccine, many people get influenza vaccines every year. Influenza is not a very pleasant disease. A lot of times people say, oh, I had the flu. Maybe not. Flu's pretty brutal. Maybe you did, but anyway, it's a pretty brutal illness that you'd really rather not have. And so it's good to get the flu vaccine. It's cheap, it's available, it's harmless, and it's sometimes effective. It's not as effective as maybe we would like, but you can prevent the flu from getting into your Christmas life. All right, let's talk about some more things that we might need to know if you really wanted to know in more detail how to do a project like we were talking about, recombinant DNA technology project or genetic technology project. Where can we find a gene that will do something we want it to do? Sometimes you can just choose a gene like the insulin gene. Sometimes you want to create a mutation, a change in a protein product or a gene that therefore will produce a protein product. And so we want to introduce mutations. random mutations can be produced by what are called shotgun approaches. Shotgun mutagenesis, in which we take a mutagen and just hit some organism with it and let it make random mutations everywhere and isolate all the different possibilities until and then screen for one that has now a protein that we're interested in having as a new property. Site-directed mutagenesis is much more efficient. There are technologies that allow us to choose and make a mutation in a specific position in the DNA code for a particular protein, and then we can have a controlled production of a new gene. And then we would take that gene again and put it in a plasmid and so forth. So once we introduce that gene of interest into a plasmid and then put that into a bacterium to make up a bunch of copies of it, then what are we going to do? What if you have millions of bacteria and we introduce some genes of interest into those by, say, cutting up a genome or whatever it might be? We want to find the bacterium that has our gene of interest in it. Selection is the way of doing that. Plasmids will also have a selection marker on it to allow us to grow only those bacteria that have been successfully transformed with our plasmid that has the gene of interest. So, for example, the plasmid that has our gene of interest might have an ampicillin resistance marker, it's often called, a marker. So we'll culture the bacteria that we've just introduced the plasmid back into with ampicillin in the medium, which will kill or prevent the growth of all the bacteria that didn't receive our plasmid of interest, but the ones that have the plasmid have an antidote for a marker for ampicillin resistance, and therefore they can grow. So wherever we see a colony, we know that our plasmid is present in those bacteria. That's called selection. One of the key tools for doing any kind of recombinant DNA work is restriction endonucleases. These are the coolest little scissors, molecular scissors for cutting out or cutting up DNA at specific sequence sites. There are about 230 different sites that we can choose and go to the store and buy a purified, highly active endonuclease protein from any of a number of different companies to cut our DNA exactly at a site that we choose. And we can engineer those sites right into a plasmid at specific locations. So it's really easy to cut open a plasmid, cut some genomic DNA or some source of DNA with a gene of interest, and plunk the pieces right together, as I'll show you. So we have lots of these endonucleases. Where I used to work, for example, we even had a vending machine, a freezer vending machine, where you could just take your laboratory credit card and go right over and purchase a particular restriction endonuclease that you needed for a project. Just walk right back over to your bench and get to work. So that's pretty cool stuff. So here's an example of some examples of very commonly used endonucleases by name right here. Again, there are about 2,000 different endonucleases that cut at around 230 different sites. And so here are some examples: BAM-H1, ECO or ECHO R1, and HIND3 or HIND3. Those are very commonly used enzymes. They're very efficient, very fast, and very effective. And you can see the sequences they cut at. BAM-H1 will cut wherever there's a GGATCC sequence in the DNA. So again, if we engineer a GGATCC sequence somewhere in a plasmid, we know that when we cut it with BAM-H1, it's going to open up at that spot and leave some specific overhangs. What do I mean by that? Here's a little cartoon showing a restriction enzyme cutting some DNA that has a gene that we're interested in, and it's going to cut where? At GAATTC. Is that ECO R1? Yeah, thought so. So you can see the enzyme cuts kind of in this strange offset way. We have double-stranded DNA, but it leaves single-stranded overhangs after it cuts. So this piece is what's going to come out after the enzyme has cut the backbone of the DNA on each strand. And so it has these sticky ends on there. They're called sticky ends because they can hydrogen bond with complementary bases from another piece of DNA cut with the same enzyme. So we'll cut this gene, this DNA that has the gene of interest in it, the gene source, and we'll cut a plasmid of interest. In this case, we're going to choose a plasmid. It could be any DNA. We'll cut a plasmid and with the same enzyme, and now the sticky ends will be complementary to each other. And so this gene of interest piece of DNA will align itself and hydrogen bond right with the sticky ends of the plasmid automatically. And we just need to use an enzyme called a ligase to fasten covalently those pieces together. And now we have permanently inserted that gene of interest into that plasmid DNA. Now we can introduce that plasmid back into a bacterium and let it grow and produce many, many copies of it and use it for what we may. So when we take a plasmid like that and insert a gene into it, then we could take that plasmid and insert it with that new gene into, say, a human cell or an animal cell or an insect cell. That's called a vector. It's something that can deliver our gene from one place to another. We're going to deliver the gene from that original source. We're going to now have a vector that can deliver it to a cell that can express that gene and produce some protein for us, for example, to make a vaccine or to make a big cauldron of factor VIII to give to our hemophiliac patients. So shuttle vectors are just vectors that can replicate themselves in more than one species. Sometimes vectors almost universally can replicate themselves in E. coli bacteria. That's by far the most common host for amplifying plasmids, but sometimes some vectors can replicate also in a eukaryotic cell. So we insert it into the cell and then more copies will be made of it and we can get a lot of mileage out of that. Shuttle vectors. But plasmids and viruses are the typical vectors that we use to insert a gene into and then deliver that gene to another cell that can do a job for us. Here's an example of an ancient early type of plasmid that was used for cloning and for subcloning, for putting genes in and delivering to different places, PUC19. We've come a long way from those days. The plasmids that we use are far more complex typically than PUC19. That's okay. You can see here that we've inserted some restriction recognition sites, restriction endonuclease sites in here, so we can cut this plasmid open with any of these 3 and sometimes as many as 10 different endonucleases, and so that whatever source DNA we had, I can look and see, well, what sites are present there that will cut out our gene. Oh, look, there's a BAMH1 site on either side of our gene. We can cut out the piece of DNA with our gene of interest, cut this plasmid with BAMH1 and insert the gene right in there. We have a resistance marker here that will allow us to select bacteria that have received this plasmid when we eventually want to introduce this back into E. coli after we do our little insertion. Ampicillin resistance is probably the most commonly used. And so we can put ampicillin, an antibiotic, in our culture media, and it will not let any E. coli grow except the ones that have this plasmid, because it can make the beta-lactamase protein that will destroy ampicillin in the medium and let the thing grow. All right, a very common way to where are we going to get the source DNA? One way to get the source DNA is to amplify, make copies of a sequence of a genome of an organism that has the gene that we're interested in. Or we can do that by polymerase chain reaction, PCR. We can also use PCR for diagnostic purposes, for seeing whether someone has a sequence that represents a genetic disease or predisposition for a disease. We can use it for forensic purposes to find out if somebody has a particular gene that makes them a relative of someone else, or shows that they have a strain of bacteria they've been handling that they used in a malevolent way to infect someone with. So all kinds of uses. But anyway, PCR is a way of amplifying a specific sequence of DNA, making copies of it, many, many copies of it. In order to do it, we need some primers. We need to know the sequence that we want to amplify, so that we can create some small pieces of synthetic DNA complementary to the DNA we want to amplify, and we'll show you what we're going to do next. We're going to start out with a target DNA. Let's say this is our piece of DNA that contains a gene that we're interested in inserting into a plasmid. Well, we need a lot of copies of it to be able to cut it with the endonuclease and insert it into the plasmid. So let's start out with our target DNA, and we will heat it up real hot and unzip, it's called. The two strands of double-stranded DNA can be separated. We can overcome those hydrogen bonds with heat. And now we have two pieces of single-stranded DNA. We add our primers, and our primers, again, are synthetic DNA that will hybridize or stick to the complementary base sequence on either end of our sequence of what we want to amplify. You can see we have two primers for either end of this double-stranded DNA sequence, one on each strand. And then we add a polymerase. A DNA polymerase is an enzyme that can make DNA. And it can make DNA by reading an existing strand of DNA, quote unquote, and choosing and inserting and covalently attaching bases that are complementary to each one of these we see here. Remember, we said that G's hybridize with C's, A's with T's, adenine, thymine, cytosine, and guanine. So the polymerase will just let one of those complementary bases come over and hydrogen bond, and then it will zip it into place with a covalent bond. So now what we're going to have is two double-stranded copies of the original target DNA. The target DNA could be a very large piece of DNA. We're going to home in and amplify only the section that we want. And so now we have two double-stranded copies of that section that we want. Then we just repeat. Heat it up, denature, meaning unzip is another word for that, is denature. Then we're going to let the primers bind again. Let the polymerase go back to work and make copies. Now we have 4 copies and 8 copies and 16 copies and 32 copies. We just keep on doing it. We have a machine called a thermocycler, which is just a fancy heating block that can vary temperatures cyclically. We keep heating, cooling, and every time we do that, we get a doubling of the amplified product. If you calculate, if you start out with only one molecule, see how many you'll have after 35 doublings. It'll be in the billions. It'll be an enormous number. So we can just set up this machine to do this and walk away and come back after eating our sandwich, and we have billions of copies of this DNA of interest. It's beautiful. It's a magic technology. So then what? Then we're going to take that plasmid that has our gene of interest in it and insert it into bacteria. The name for inserting plasmids into bacteria is called transformation. We have a naked piece of DNA, meaning it's not in an organism, it's not part of a virus or anything else, it's just floating. We're going to introduce it into the bacteria by transformation. Or if we want to bring it into a eukaryotic cell, that's called transfection. Just two terms that we have to learn that are used to describe those two processes so we can tell them apart from each other. And they're technologically done very differently. Some of the ways that we can actually do that, electroporation is very commonly used. Give a high frequency shot of electricity to the cell for just maybe a half a second, and during those electrical discharges it punches holes in the membrane temporarily, the phospholipid membrane, and plasmids can go right in. Or we can take some cationic, some positively charged detergents and mix it with the DNA, these lipid molecules that have detergent properties, and then mix that with our cells, especially when we're doing transfection of eukaryotic cells, and the cells will end up taking up the DNA into the cell. Transduction, we can, instead of using a plasmid, we can use a virus. Insert our gene of interest into a virus and then produce some virus particles first in a packaging cell line, and then take the virus particles and transduce some bacteria or eukaryotic cells. Protoplast fusion, I won't make any bones about that. That's just another method of introducing DNA. All right, we got enough to worry about. So here's a little cartoon showing 4 ways of inserting DNA into a cell of interest. Electroporation, they drew the discontinuities in the membrane, right? We're using an electrode to shock this cell with electric current, and it produces some temporary holes in the membrane just instantaneously, and plasmids can get right into there. Lipofection, people have taken the word transfection, which means to introduce DNA into a eukaryotic cell with lipids, and they made up this word lipofection. That's where that comes from. So we're going to mix cationic lipids, detergent-like substances, long-chain fatty acid-like things, with our DNA and let it go into this right into eukaryotic cells typically. Microinjection, there's a cool technology. You can have right on the lab bench a machine that can, while you watch under the microscope, manipulate a microscopic glass needle and just stab it right into a cell. You're controlling a joystick that's moving microscopic things under the microscope and stab right into the nucleus of a cell, give a picoliter or two of volume, shoot right out of that needle, and then pull back out again. And now you've just injected DNA right into a cell. Now that's pretty cool stuff right there. Microinjection. I hope you'll notice this K right here. Um, this particular graphic was produced by somebody in a country where they don't use the letter C for a hard consonant C sound. In their words, they use Ks like a number of Germanic languages. That was cute, I thought. So this is just, I guess I could have cycled through some little text that was there. Electric pulse for electroporation. Liposomes are what you form with these lipids mixed with DNA and it goes into cells. Microinjection um, and viruses. Viral transfection is called transduction. It shouldn't have said viral transfection. It should have said transduction. We don't say viral transfection typically. All right. Another way DNA, other ways, microinjection we just mentioned, a gene gun. Sometimes when it comes to plant cells, if you're trying to modify plants, and there's a lot of progress that can be made in producing improved agricultural species by using the gene gun. You can take a shot of high pressure gas with some heavy nanoparticles coated with DNA, like gold particles, and shoot them at super high velocity at some plant cells, and it'll just shoot right through the plasma membrane, and you can produce some genetically modified plants that way. But we won't talk much more about plants. There's a picture of someone using a gene gun. This is a gene gun apparatus. There's a tank of gas that can under high pressure that's going to be used to propel some heavy nanoparticles into some plant cells that are here in this dish. We have such a gene gun if anybody's interested in a project. We have a gene gun with no one to really use it right at the moment. The faculty person that was interested in that instrument is no longer here at Roberts, but it's there, and it might be a fun thing to make some genetically modified plants as an experimental thing. Here's a picture of microinjection about to happen. I'm going to shoot a little burst of DNA containing liquid into a cell nucleus. All right. So obtaining our DNA of interest to insert into a plasmid or into a virus vector. We need some primers if we're going to do PCR, or we can make a genomic library. That's a whole nother way to get at getting a gene of interest so that we can use it for our purposes. A third way that I want to tell you about is probably my favorite way of all. You can take messenger RNA from a cell that you know expresses a particular gene and then work backwards and make a DNA copy of that messenger RNA, and then we can insert that right into a plasmid, or we can amplify it by PCR and then insert it into a plasmid. So what about making a library? We can take a genome that we know contains the gene of interest and cut it right up into pieces using endonucleases, right? Just to make some workable sized pieces, not so small that we're going to cut our gene of interest. We're going to have at least one copy of a piece that comes out with these restriction endonucleases that contains our entire gene. Insert the whole different, just gamish, thousands of pieces from that genome potentially into host E. coli bacteria. And then we're going to let all of colonies grow of all these different E. coli bacteria and screen them for the one that has our gene of interest. And then we'll take out and make a pure culture of that. We'll take out a single colony and grow it at a large quantity of it. Now we have our gene of interest in a plasmid. We can purify that plasmid. So that's called a library. We wind up with about thousands and thousands of bacteria that all contain different plasmids with different pieces of this original genome. That's called a library. On a different day, we might say, gee, we would like to isolate a different gene from that same genome. Well, now we have frozen down in our freezer a library of thousands of different E. coli, each one of which has a different piece of the genome in it, and we could do a screening and choose a different um, um, clone, a colony to pick for our purposes. That's like a library, a genomic library it's called. A different way to make a library is to insert all the fragments into bacteriophage. And so now each different phage, these are just viruses that are specific for infecting bacteria, each phage now has a piece of the original genome in it, one of which undoubtedly would have our gene of interest in it, and then we can use those to infect bacteria later. So we could have a phage library. Again, we keep it right in the freezer, amplify it up to so we have a large number of copies of every single one of the different phage that had different pieces of the original genome in there, and keep that right in the freezer. And when we want to when we want to isolate some particular gene, we can just infect some bacteria with phage, and so actually we can do it right on a huge plate. We can infect a lawn of bacteria with phage, and then screen for a place where there's the gene of interest is present, and then we can isolate out that particular example. Anyway, just some notions of how we can get the gene of interest that we want to insert into a plasmid or a virus. Again, my favorite one, take like maybe some human cells. If we knew that there's a human gene that we wanted to express, such as a clotting factor, you can't use the genomic DNA directly to express a clotting factor because there's introns. Eukaryotic cells put introns in the coding sequence for a particular protein, and so if you tried to express that directly, it wouldn't work. You'd have all this non-coding DNA, chunks of it, all sprinkled in throughout your codes. That won't work. What we'll like to do is take the messenger RNA from the cells while they're expressing that particular gene of interest and purify that messenger RNA, and then use a reverse transcriptase enzyme that comes from retroviruses. So now we isolate the messenger RNA from the cells that we know make the clotting factor, for example, and then we will insert or make a copy, a DNA copy of that. This purple line represents making a complementary DNA to that messenger RNA for the clotting factor, for example, using a reverse transcriptase type of enzyme. So DNA strand being synthesized, and we'll simultaneously chop up the piece of RNA, and then we can make a double-stranded DNA out of that, and then now we have a copy of our gene in just the coding sequence. Now when we insert that into bacteria, it's ready to make protein out of it, or into a eukaryotic cell. No introns. So it's a fast, easy, and beautiful way to get a gene of interest, and so it's very commonly employed. You can actually, if you have a short enough sequence you're interested in having, you can just synthesize the thing from scratch. That's how we make primers anyway, in order to do PCR. Well, you can actually make a relatively long, it's gotten so efficient, you can make a relatively long piece of DNA, error-free, and has to be tested and screened, of course, but you can make a pretty long piece of DNA, and say a short little gene like that would produce an insulin, no problem to synthesize something like that. I thought it was funny to see this picture because here's sitting on top of this DNA synthesizer. This is a rather old picture. It's a dot matrix printer. I don't know if any of you guys are much too young to know what a dot matrix printer is, but if you've ever heard of, it's like an inkjet printer. Inkjet printers are still around, right? You have this little head that has like hundreds of little nozzles or dozens of little nozzles that can shoot microscopic droplets of ink in a pattern as it passes across a piece of paper, and it can leave behind characters, the shape of characters. Well, before inkjet printers came along, there were dot matrix printers that instead of having little nozzles had little wire hammers that could be actuated by little solenoids, and as the printer head went across line by line across a piece of paper, you'd hear this loud sort of screaming noise as the little pins would be stamping along and making rough characters. And it was quite something to behold if you've never seen it. And it took forever. Each line of a piece of paper had to be printed zip, 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 and you could make a whole page full of text. But now we can make 30 pages of text per minute on a laser printer. So we were like, what? But that's just what you see on top of that. That was just a thing not that long ago. At least to me, not that long ago. To you, probably infinity ago. 1990s, still inkjet printers existed. So, we talked about selection. We said that when we insert a plasmid back into bacteria in order to grow it up for us, get a clone, a clone, a population of cells that contains our plasmid with our gene of interest. How are we going to know which bacteria has a particular gene of interest? Well, one thing we can do is use ampicillin in the media and have an ampicillin resistance gene in the plasmid. That's good. We want that, so we only have down here growing plasmids that contain our bacteria that contain the plasmid. But not all of the plasmids, when you insert a gene into it, not all the plasmids in your test tube are going to successfully receive your gene of interest. So we're going to have another way to narrow down even further, not just colonies that can grow on ampicillin, colonies that contain our gene of interest or contain a gene. And so this particular technology involves using beta-galactosidase gene for selection. So what that means is this plasmid has the beta-galactosidase gene, which can take a sugar, a modified sugar, and convert it into a colored product. And so if we just took this plasmid, inserted it into bacteria without having done anything to it, and plated it on media that contains this special modified sugar, all the colonies would be bright blue. A very cool thing to behold. Then, if we insert our gene into a restriction endonuclease site, we're going to put that site right in the middle of the beta-galactosidase gene. Is that interesting? So if we successfully insert our gene of interest in here, it's going to separate the beta-galactosidase gene into two parts. It will not have activity. It'll no longer be able to produce that blue color, and so we'll see white colonies. That's called blue-white selection. It's still used to this day. It's a very powerful, quick, easy way to find colonies that have successfully received some DNA during our insertion step. And so now we can pick the white colonies and screen them more carefully for the specific sequence and make sure it's got our gene of interest in it. Another way, another piece of technology we can use is colony hybridization. If we had a whole bunch of colonies growing on some medium, agar, for example, we could take a special kind of paper that will stick nucleic acids onto it very avidly and lay that right down over top of these colonies and then pick it up again. We have it in the presence of a nasty detergent that will pop open the bacteria, and the plasmids will come out and stick to the paper. So wherever there was a colony, whatever plasmids were present is now stuck onto this paper in an identical pattern. We can mark this with a special ink so that we can see exactly where it came from on this plate. And then we can probe it. A probe is like a primer, only it has some kind of a tag on it to make it visible. So we have a short synthetic piece of DNA that's complementary to our gene of interest. And it's got like either a fluorescent dye on it, or it might even have a radioactive, a little radioactive atom on it. And then we can let that hybridize, and it will only stick where there's a complementary sequence. It'll only stick to the colony where there's a clone of bacteria with our gene of interest on there. And so then on the paper we can say, aha, this colony and this colony right here have the gene of interest in them and no other ones. And because we know exactly the orientation, we had that originally on the plate, we go back to our original plate that still has live bacteria on it and say these two colonies, we're going to pick those out, take a sterile applicator and take out those two colonies and grow them in some fresh media. And now we have a pure culture of our bacteria that contains our plasmid with our gene of interest. So that's another way of selecting. Instead of blue-white selection, we can use this method, this probing method, to do it. E. coli are commonly used for actually, once we get all that done, and we now have bacteria with our plasmid, with our gene of interest, we can now let those bacteria produce a protein for us, for example. And that's very easy and cheap to do. And it's wonderful. We know a lot about the bacteria. We can make them do what we want, a lot of properties we can control. Um, One downside of it is that um, the bacteria will not secrete a product. Let's say we grow the bacteria in a huge cauldron, a huge vat in the factory, and we want to make a product to sell to give people the medicine they need, either insulin or a clotting factor or a vaccine. Well, we're going to have to do something complex to break open the E. coli in order to get the plasmid out, or get the protein out, I'm sorry. And the problem with that is E. coli are gram-negative bacteria. So they have terrible lipopolysaccharide on their outer membrane, which is very, very toxic to humans. So we have a big cleanup job to do to get rid of all that endotoxin, it's called, from the whole process. So that's a bummer. And but it's still, for a lot of purposes, a lot of situations, it's the way to go because it's so cheap and easy to use E. coli to grow up and to express a particular product. Another way to go, though, is to use yeast. Saccharomyces cerevisiae is a very commonly used yeast for both for research of all kinds on eukaryotic cells, but also for producing products that we're interested in. It doesn't have any of that toxicity to it. And we can have yeast that will secrete a product. And so we may be able to just have a big vat of yeast growing, and out comes all the protein that we're interested in having. We can easily separate the two and purify the protein. Plant cells, whole plants can use to express products. Mammalian cells or insect cells can be used to express products. They're a little complex, a little more expensive. You can't grow them up as easily in as larger quantities, but the beauty is that you can produce complex protein products that need to be folded up in a specific way that only eukaryotic cells can know how to do, or maybe making all kinds of disulfide bonds in specific orientations, all kind of good reasons to use more complex cells to make a product sometimes. So we've come a long way. You got a lot of good ideas of how to make some things that can really help us in our lives. So we can make enzymes, as we've said. We can make vaccines. A subunit vaccine is just a vaccine that's made up of one part of a virus, for example, one protein that came from the surface of a virus. That's called a subunit vaccine. We just purified it in the laboratory. Gene therapy, we can use viruses that have been modified in the ways we've been talking about to introduce into people's bodies to now deliver a gene that they're missing, or that they have an inappropriate copy that doesn't produce the right product. We can now introduce viruses into their body so that their own cells will now produce a healthy gene product that will cure a disease. So there's a number of diseases that have been effectively treated by gene therapy. One way to do gene therapy is to have cells viruses that will cause cells to express a copy of a gene that's now good, whereas the patient didn't have any. Or another way to do it is to inhibit the production of a gene that we don't want the patient to make, that's inappropriate, that's causing disease. So we'll just introduce one more DNA technology: small interfering RNAs, siRNAs. They're a type of RNAs that will hybridize with a messenger RNA. And then once that hybridization takes place in a cell, eukaryotic cell, it sees this double-stranded RNA, it says, oh, trash, and it will send around a protein complex that was designed to break down that messenger RNA and get rid of it. So we can take away messenger RNA from a particular cell by introducing specific sequences of small interfering RNAs. So in this case, we can use our genetic technology to produce a virus that will deliver the sequence for siRNAs into the host cell body, and then their cells will start producing siRNA and inhibiting the synthesis of a troublesome protein by getting rid of its messenger RNA. So that's another way of approaching genetic or gene therapy. Oh, there is another technology that we might as well throw out here because since we're on the subject of genetic technology, relatively recently, this method was developed, which allowed us to sequence the entire human genome. Now that's a lot of base pairs. This is in the billions of base pairs. How do you sequence billions of base pairs in all the 46 chromosomes of human cells? Well, you do it by this shotgun sequencing method. First, cut the genome right up into fragments, the entire genome into fragments. It doesn't have to be the human gene. It could be any species we want to sequence whatsoever. Then we're going to clone all those pieces, just like we talked about before, into a plasmid. In this case, we're going to introduce pieces into individual bacterial artificial chromosomes. They're called, a little bit bigger than a plasmid and a little more complex than a plasmid. But again, we're going to have thousands of copies of these artificial chromosomes, that each of which has a separate piece of the original genome that we want to study, and then we can just sequence the insert in each one of these chromosomes relatively easily, these artificial chromosomes. And there's biochemical methods for doing the sequencing relatively easily in the lab. So now we have thousands of little mini sequences that we've gotten from all the fragments of the genome, and there's wonderful software that's now been developed to analyze all those sequences and see where little bits of them overlap and how they must have fit together and produce the entire original genome sequence. And it's just a miraculous thing. So now, again, the Human Genome Project allowed us to sequence the entire human genome many, many times over now. And so we're learning a lot about the variability in the human genome, various genetic diseases and things like that by just studying more of the human genome. That's shotgun sequencing has made that possible. So yeah, we have the sequence of multiple individuals, many individuals now. Proteome project. Proteome means all the proteins that are produced by the genome, and so there's a lot of interest now in studying the proteins that are expressed in a cell or by a particular organism and so forth. So we won't go too much more into that. But okay, I think we're getting down to the end of our chapter. DNA fingerprinting we'll talk about briefly. DNA fingerprinting is a strategy for looking at small differences in nearly identical DNA. We'll take the genome from two different sources, say, two different individuals, cut them up with restriction endonucleases into relatively largish pieces, and then we'll separate them on a method called gel electrophoresis. We'll take effectively what's like an agar gel that we would grow bacteria in. We'll use a little bit more pure agar that's clear. Agarose it's called. And we'll just insert the chopped up genomes of different individuals into little spots. Like there's this represents a gel that was placed in an electric field, and we just pipetted, dropped a few droplets of liquid with the chopped up genome of different individuals into little slots or wells, they're called, in this gel, then set up an electric field so all the pieces began to migrate down towards the positive electrode, and they migrate at speeds that vary by the size of the fragment. So all these little bands, then we use a stain to make the DNA show up. All these little bands, they're called, represent fragments of different sizes that came from those original genomes that were cut up by the same exact endonucleases, and we're looking for something unique. And so in this case, we're looking for some E. coli that was contaminating some juice that a bunch of people drank, and we want to see whether a particular E. coli that has a toxin that we know about is present. Sure enough, some telltale bands you can see right here and right here that are only in the E. coli isolated from people who got sick by drinking contaminated juice. All these lanes right here, and you can see that those unique bands that are not present in the control samples. And here's some apple juice that was potentially the source of the sickness. And sure enough, the E. coli was found in that apple juice and has the exact same bands that were unique to the people that got sick. So you can see that we've been able to use DNA fingerprinting to show that a particular juice sample was what made a bunch of people sick that all drank the same juice. So very powerful strategy for public health. Southern blotting is a way of identifying a particular gene or sequence of interest that's in some DNA by using probes, just like we talked about before. We talked about using probes for making a replica of a bunch of colonies on a plate. In this case, instead, we're going to cut up a genome, much like in this method. But when we separate all these fragments, instead of just looking for a unique band, we're going to use a probe, right? We're just going to take some genome, cut it all up into pieces, separate all the pieces on an electrophoresis gel, and then we're going to transfer all those bands of DNA, those fragments of DNA onto a paper, just like the nitrocellulose we saw in this example. Where'd it go? I guess it's too far back when we did the colony replicas. We transfer from the gel on. It's hard to use a gel for doing identification processes, so we're going to mash the gel down with a bunch of weights and crush it down and let all the DNA diffuse over onto this sticky paper. It's not sticky to touch, it's sticky to DNA. And we're going to cast. So all the bands are now here. We can then use a probe, again, with a synthetic piece of DNA with a specific sequence with a fluorescent dye on it or something, or a radioactive tracer on it, and identify the band of interest. So that's just a southern blot, it's called. Let's say, I don't go too far afield, but let's say we have a bunch of genetically modified animals that we're using in the research lab. Whenever we're about to make a new generation of baby rats, for example, or baby mice usually, that are these genetically modified mice that we're going to use to try to understand the function of a particular gene that's been eliminated or modified. We constantly have to screen the colony to make sure that the mice are still carrying the gene of interest. We'll do a southern blot. We'll take a little sample from the mouse body and purify some genomic DNA, cut it all up into pieces, separate it on a gel, transfer it onto this paper, and voila, we can verify, yep, our alteration that we've made in the genome of those mice is still present by using southern blotting. Forensic microbiology is a huge huge area of application of genetic technology. Same as exactly we were talking about just now about that DNA fingerprinting for the juice. One thing we can do is use reverse transcription PCR or real-time PCR. These are just modifications of PCR to allow us to amplify lots and lots of cDNA from the cells of people's bodies in order to compare with each other. We can use those, for example. You might try to demonstrate that the semen that was found on a rape victim was an exact genetic match for someone who was a suspect in the rape case. There's a whole bunch of really interesting examples. If you take Introduction to Forensic Science at Roberts, you'll learn about a whole bunch of different examples of where genetic technology was used to solve cases where people did bad things, like a doctor who injected his girlfriend. He wanted to get rid of his girlfriend. What did he do? He injected her with human immunodeficiency virus from some samples that he had from patients in his laboratory. You can't believe it, right? Happened. And genetic technology, as we've been talking about, was used to verify that the exact sequence, all viruses tend to mutate quickly, the exact sequence of the HIV genome that was found in this poor woman was the same as a sample that was found in the doctor's office and that had obviously been harvested. Some had been taken out of it, and they were able to convict this physician. Anthrax. There's been, I hope you've heard of some cases in which a person that worked in a particular laboratory that worked with anthrax, which is a type of bacteria that can cause lethal disease in humans, grew up a bunch of anthrax and caused it to produce some spores, which then were delivered to U.S. congresspersons in the mail in order to create a shock. And it did create a shock. Some people were killed, and genetic technology was used to identify that the strains of anthrax that was sent in the mail was identical to strains of anthrax from a laboratory where this particular person worked, and was able to show that they eventually were able to convict this person of having poisoned other Americans by sending it through the mail. All they had to do was open the letter, and a little cloud of these microscopic spores would come into the air, and they would inhale it, and that's all it takes, because inhaled anthrax spores are so dangerous that they would kill a very high percentage of people that were exposed that way. So craziness. A norovirus outbreak. Let's say you were on a cruise ship, or you got ate some contaminated food and got norovirus. Well, there's an example of when public health was served by using DNA technology. Um, I don't want to go into detail because I've been talking too long already, but suffice it to say, 100-something people were found that had gotten sick, complaining of the same symptoms in the same community at the same time, were all sampled, and their DNA was examined, or their feces was examined, I should say, stool samples were examined by reverse transcription PCR amplification, and found to have a unique banned product from digesting down the bacterial DNA and looking for a particular 213 base pair PCR product that's diagnostic for norovirus. And they found that all these people had the exact same product in their stool samples. And here's that 213 base pair product. These are all the patient samples here that you see on the right. These are the control sample. There's a control sample on the left. It is not infected with norovirus. So they then sequenced actually these products to make sure that they actually was in fact norovirus, and they were able to identify the source of the norovirus as a particular as some contaminated lettuce that was contaminated by a particular worker at a prep cook at a restaurant that had produced sandwiches that all these people ate. So I mean, we've had, now this is our third example, but you can see the power of this technology for solving human public health problems in real life. There are some ethical issues associated with genetic technology. For example, if someone's genome is sequenced for some reason, whose property is that? And should the government have access to that information in order to maybe serve the public health in some way at a later time? Or is that private? Or should a company who does the sequencing be able to hold on to that in order to do some research on it and use it for developing some products in the future? There's a lot of ethical issues that need to be ironed out with respect to DNA technology. But we better end there, and I invite you to come back and join me for the next chapter, which we'll be doing relatively soon.